Hello, my name is Viktor Jirsa. It's a pleasure to speak to you on responsible full brain modeling for clinical translation. And essentially, what I want to express with this is I want to give you an introduction into full brain network modeling using the virtual brain, dealing with non-invasive brain imaging data, providing a constraint upon brain models that allow actually to make them patient-specific, and this is a basis for making the translation into clinical applications. The expression responsible shall actually indicate that this has to be done very carefully because there are consequences, and hence very good diagnostics is needed for this. So this is our ambition. We wish to understand the brain through personalized brain models and through the building of patient-specific or personalized individual brain models, sometimes referred to also as digital twins. I'll speak more about this later. There are challenges. Whenever we do neuroscience, we are interested in disentangling competing mechanisms. What this means is the brain is a multi-scale nonlinear system, and there are multiple causes that can give rise to the same type of phenomenon. We refer to this as neurodegeneracy, and this is a natural consequence of the multi-scale architecture of the brain. And there are good points to this. Namely, when you have neurodegeneracy, what this means is that there is a propensity of generating the identical behavior by using completely different parameterizations or mechanisms. This is good when we are having a situation where the brain receives an injury or any type of perturbation. So it can actually adapt in order to reparameterize itself to compensate for the injury received and to perform the same type of behavior. From the scientist perspective, it is a nuisance because it evokes competition of individual mechanisms. There is a heterogeneity of different mechanisms giving rise to the same type of behavior, and that renders them non-identifiable. Yeah? That's one very important issue with its positive parts and its negative parts. Apart from this, we have an aspect in the context of personalization that we cannot work only with in vivo data, which are necessary for personalization, but in order to achieve higher resolutions, which actually improve the predictive power, we need to use also ex vivo data. So this is a framework in which I want your mindset to be. And as a first application, I would like to walk you step by step through the construction of what we refer to as the virtual brain, which is actually a neuroinformatics brain simulation platform that allows you to perform this type of brain simulations, but very often this type of brain models are referred to as virtual brains because of the simulator's name. So what is a virtual brain? Essentially, you are starting from the measurement of non-invasive brain imaging data of an individual. And typically we do this with MRI. You extract the topographic information, which is cortical surfaces and the subcortical subsystems. You parcellate it, which provides us the nodes in a network, but using diffusion-weighted tensor imaging, so diffusion MRI, we can actually extract the tracts, the individual tracts, the so-called connectome of an individual's brain, and we can actually co-register this and organize the tracts of the connectome such that they refer to the actual nodes in this particular network. So we end up with a network with a particular topology, left hemisphere, right hemisphere, and with a parcellation specific organization. When we unfold this and span it in a three-dimensional physical space, we have actually an individual avatar. An avatar is a geometric form, but there is no function in there yet. But it spans it in three-dimensional space. In order to get the function in there, to render it functionally useful, we need to equip it actually with mathematical models. And this, on this level of organization, we do using so-called neural mass models. These are models where we use techniques from statistical physics, in particular mean field theory, where we use microscopic representations, typically point neurons that we cluster in individual populations. Typically, we put them in excitatory and inhibitory populations. We separate them. And then we apply these techniques coming from statistical physics in order to extract so-called collective variables that are representing a information compressed representation of the activity of the neurons being cast into mathematical equations, so you can actually run them autonomously in order to generate the activity of the population. So typically this gives a dimension reduction from several hundred of thousands to 2, 3, 4, 5, 6 dimensions, something like this. There we have absorbed in the individual parameters the properties of the various neurotransmitters and neuromodulators, in fact, AMPA, NMDA, GABA, that are providing parameterizations of the excitatory and inhibitory population. Examples of this type of models are listed here. This is coming from the lab of Alain Destaix, where so the citations are down there. Yeah, this is an example from Divolo et al. In fact, a simulation of a network of a few thousand inhibitory and excitatory cells under different parametric conditions. You see here intended to model sleep-like spindle states in slow oscillations in sleep, and wave-like activity representing asynchronous states that we have present in the awake state. When collapsed into the collective variables, there you can actually mimic via the reduced mean field equations the behavior you see of the collective variables, which are actually then the meaningful quantifier that we use to characterize the activity in an individual network node. And those nodes can actually then be studied mathematically. You see the difference in the types of dynamics, and their parameterization can span a parameter space in which you then can find actually characteristic parameter settings for the individual states. Put this together back into the virtual brain. You have a differential equation with a location x at every single network node location in the brain, and evolving over time. This is the nonlinear dynamics of the neural mass, nearest neighbor connectivity, a local dynamics if you have very many vertices, plus the communication via the connectome-based connectivity skeleton communicating between far distant areas x prime, projecting onto the area x. This is what the mathematics is behind that. So essentially the following is happening. From non-invasive data, we extract the topography and parcellate it in network nodes. Then we replace each network node by a sphere or a cube, in this case, representing the population of the mean field activity of neurons. So we replace it by the neural mass and then connect the neural masses by the individual's connectome. And then this represents the source level. Then we can actually put in measurements such as sensors, as we do in epilepsy, so-called stereotactic EEG. These are needles that we insert into the brain of a patient suffering from epilepsy in order to explore pre-operatively the organization of the activity, or non-invasive approaches such as MEG or fMRI. So we can actually map from the source to the sensor activity, which is another form of personalization, because the sensor organization, like this implantation scheme, here are also patient specific. Like this, we can go all the way from mathematics to the brain imaging data, the way you measure it actually in the clinic. Here, I just want to point out that the virtual brain platform is available under this website here. We have pretty close to 40,000 users today, and it has a graphical user interface. It's open source, it runs on Python, and you can essentially define the neural mass models here. You can pre-process the data for the connectome and integrate them in the platform of the network model. You can manipulate the data, and then you can run the simulation, either individual simulations directly on the source level, or large batch jobs when you're doing large-scale parameter searches, parameter sweeps, in order to obtain the type of parameter investigations that are necessary, because the parameters are the interface between the math and the actual patient data. Here you see, for the same source dynamics that has been simulated here, the EEG data of this particular patient as it is typically recorded in the clinics. So everything I will speak about can actually be reproduced in this platform, the virtual brain, which is integrated in an even larger workflow available on eBrains, the European infrastructure for digital neuroscience. You will see more about this a little later. So what I wanted to show you is a very specific example, namely using the virtual brain applied to questions in epilepsy. Epilepsy is our workhorse. It is the most advanced in terms of virtual patient modeling. There are other applications in terms of Alzheimer or Parkinson's disease, as we have heard before, in neurodegenerative diseases, multiple sclerosis also. Often they are serving as a diagnostic biomarker. If therapies, like in the case of brain stimulation, exist, then this can be perfectly modeled in TVB being the virtual brain, so that is a platform of choice. A few words on epilepsy to provide you with some background. It is one of the major brain disorders from which human beings are suffering. 1% of the world population is epileptic at some point in their lifetime. 60% can actually treat it pharmaceutically, which means 30 to 40% are not treatable pharmaceutically. And for them, the question raised is essentially how to treat them therapeutically via surgery. So brain surgery is the second best next option. And for this, actually, you have to explore the brain's activity organization before, during, and after a seizure. One way of doing this is with invasive electrodes, so-called stereotactic EEG, because they enter into the volume. They're not just on the surface, but they actually enter into the volume, hence stereotactic, which allows you actually to go also to deeper structures, subcortical structures, which are often implied, and then record this type of activity, allowing you to analyze the data. There, please note the success rate of epilepsy surgeries around 50 to 60%, as you see here. For the temporal lobe epilepsy, 60, 65, but it drops down all the way to 40, 30% to extratemporal epilepsies, in particular frontal epilepsies, 25, 30%. As surgery success rate has not changed over the past 30 to 40 years. So there is a need for help. And the workflow we have developed is the following, which is inspired by the virtual brain, but targeted towards the use in the applications of epilepsy. So from the non-invasive data, as discussed before, we compose the virtual brain avatar using topographic and topological data. span the network in 3D. We equip it with mean field techniques, so it becomes functional. We can refine the network model by taking information available in the MRI, for instance, of the patient that helps us to constrain the parameters. And then one way of using this such personalized virtual brain is you can actually fit it and ask questions: where is the epileptogenic zone? Characterized by the onset of seizure activity that then propagates and recruits other brain regions. So this becomes, in this case, the virtual brain becomes a template for data fitting, providing an estimation of the epileptogenic zone, which then is being passed on to the clinicians, and the patient is undergoing surgery. There is, in terms of the mean field dynamics, there is a large literature of papers available, and I simply wish to point you into this direction of the literature by citing two very relevant papers here, in this case, on the nature of seizure dynamics, in which we have actually characterized the dynamic properties that a mean field model has to have in order to be able to perform epileptoform the dynamics. I'll show you a few more details here. Yeah, this here is a seizure as recorded from the rat with an onset, discharge, offset. Look at the scale here. These are hundreds of seconds. Yeah, the fast discharges, you see them here. In parallel, a recording has been performed. This is a hippocampus in tuto of the extracellular potassium, and you see it actually traces out the seizure dynamics here. During the seizure, jump off, seizure onset, jumps up. So extracellular dynamics provides an envelope function for the discharge dynamics. Here you have a few more details. So you have the seizure train, extracellular potassium, and here one more time zoomed in, and here you see more details of this. Onset, discharges, starts jumping, tracing out, and returns to its origin. Whenever you have a dynamics like this, mathematically, I just want to give you the gist of this. You can actually split the dynamics that you have seen here in a fast, slow time scale. Here, slow, fast, slow, fast. This allows us to evoke a set of theorems that actually allow to separate fast variables from slow variables, and they always must exist when you have this type of dynamics. You can actually unfold them in a state space where this is a slow dynamics, this is a fast dynamics, And what you see here in terms of seizure train maps actually upon a mathematical object, which is a spiral here. This is a slow dynamics, and then you undergo a bifurcation where the resting state dynamics destabilizes and you go into the seizure state at a different location at the state space, then you discharge, and then the seizure stops and you jump back over here. This is a slow dynamics, which would actually correspond to the extracellular potassium. And the fast dynamics would be the local feed potential and its first time derivative. This provides you exactly with this type of spiral. And actually this here is an experimental data set from the in tuto hippocampus I showed you. So this is slow dynamics, fast discharges, etc. So the geometry of epilepsy is actually this spiral, and it is characterized by this entry and exit bifurcation that allows you to actually go into the seizure state and outside the seizure state. Mathematically, you can actually go in more depth there, perform bifurcation analysis. So bifurcation is a transition, the way how a system reorganizes itself. It can be actually classified when it undergoes from one state to the other. And there is only a finite number of ways how this can be done. And in this particular case, there are actually only 6 planar bifurcations, how a system can go actually from a seizure state to a rest state or from a rest state to a seizure state. You can actually characterize them. You can write them down mathematically, can characterize its amplitudes, its frequency, and they are generic. They are so-called canonical behaviors. For each of them, we have a mathematical representation, and when we put them together, we get actually the spiral structure I showed you, and it allows us actually to build a taxonomy in terms of onset bifurcations and offset bifurcations in terms of 16 cases, so-called dynamotypes that exist and can actually be theoretically predicted. And this paper here in eLife by Sergio et al. Is a beautiful statistical analysis of hundreds of different, actually thousands of different seizures from hundreds of different patients collected in more 10, 11 laboratories worldwide in Australia, Japan, Europe, the US, and actually characterizing this and validating the existence of this type of taxonomy and identifying two leading classes that are characterized by two very specific sets of onset and offset bifurcations. And here you see two examples of them, actually, the two leading classes. These are human experimental data where the onset happens. You have a jump, fast discharges, and a slowing down towards the end, and seizure offset. This is a characteristic saddle node bifurcation with a baseline jump, and then showing characteristic behavior towards the offset at the seizure end. So this can be done systematically, providing us with a mathematical skeleton that can be imposed upon the mean field dynamics, allowing us to parameterize these type of models. So it's a very powerful way guiding our thinkings, but it's making also other predictions, such as you can actually analyze these classes, and you can actually, can we predict the most prominent seizure types, the dynamotypes, simply by analyzing this bifurcation patterns in more detail? And the answer is yes. Yeah. So you can actually go and analyze them in terms of the complexity. How many parameters do you need to tweak in order to generate a particular class? And the more parameters you need to tweak, the less likely it is actually to be robust against perturbations, etc. So you can actually predict the most in this taxonomy of 16 classes. You can actually predict the most robust and prominent dynamotypes to be found, and this is actually nicely validated by what has come out of the classification in the individual seizures empirically. It does not correlate though in any time with the clinic. Yeah? So that was kind of disappointing, because we had hoped for this, but this is not the case. But it follows at least the scheme of the complexity as observed in the dynamics. So now we have a very well characterized and detailed neural mass model for epilepsy. Now we need to put it to a use. And actually, we need to extract the data feature. We can actually use the core features or the pure features in the raw data with all the discharges. We have not done that. In order to take a more simplistic approach, we have actually focused on the seizure envelope. So you have the preictal spikes, then you have the seizure onset at this location, and then you have the fast discharges until, and then the slowing down towards the end, and then the seizure stops. For a large variety of seizure types, as you see here, you can actually compute the seizure envelope, or here you see a frequency time spectrum for this type of time series, and you see actually that the fast discharges providing a nice envelope for the evolution of the seizures. We have chosen this as our data feature that we wish to extract and actually fit. In the workflow that I showed you earlier, there was a fitting process. Yeah, we can do this. And what we're using is actually Monte Carlo techniques, because they have the advantage, specifically Hamiltonian Monte Carlo advantage, that they are not just optimizing parameter, providing you with a single simple set, but they are actually sampling the range of parameters that is possible in the spirit of a neurodegenerate system, trying to capture the whole range on the range of brain dynamics that can be explained by the set of parameters being capable of generating this. So we take the mathematical object, when we project it, so the spiral, and project it into the plane, so project down on the slow variable, then we actually get a projection here. This is a resting state. And when a seizure occurs, you leave the resting state and you go in the spiral, but in the projection, essentially, you deviate on this side and then you hang out there and then you go back. This is actually experimental data. We can actually sample them for different network nodes. These are the so-called chains, that the Monte Carlo chains that we run and sample the parameters. So two nodes that don't seize, these two here seize, and this one does not seize. So we can do this systematically, and it is actually, I must emphasize this, it is a must. It needs to be done because of the neurodegeneracy in the system. And these here are actually experimental data. When we sample the excitability of the system, there is also a degree of non-identifiability that cannot be ignored. You need good diagnostics in order to make actually responsible judgments about your degree of ignorance or your degree of confidence you can have in these estimated parameters for this one individual. Here you have an experimental data set. In blue, we did this with Monte Carlo Markov chain, and this is the excitability. For here, we are actually in the epileptogenic zone areas. And here, this would be more healthy activity, but you see it's actually multimodal. And this is a posterior distribution of the excitability that was sampled via the Monte Carlo technology. And you see actually that we do not have a very narrow shrinkage to a single value and area, which would be the ideal case, but that we have to take actually non-identifiabilities into account, because what Monte Carlo is doing, it's actually balancing the compatibility of the model and the causal mechanisms that are inside of the model against the data, and trying to estimate the confidence via posterior distributions that we have here. If you now take some other approaches, for instance, a Gaussian approximation, then you will get essentially just garbage out, because it is incapable of estimating or dealing with such a distribution. It's better when you take approaches where you, for instance, estimate where is the center of mass of this. This provides a nice element there. Always a maximum. Yeah. So looking for the maximum a posteriori. These things we do, and we put them together with multiple nodes, and then we get this type of landscapes of modes that we need to deal with. And this is essentially the work in the estimation. Just to show you that these things are actually meaningful, we can actually then estimate the excitability for a whole brain network for the same patient, where we are actually taking, in one case, ignoring the connectome, which means this is a model feature. And in this case, we take it into account. And in green, you see the epileptogenic zone, which provides actually different areas for the left fusiform gyrus here than for the case where we take the structural connectome into account. And there are also other differences. So we are plotting this as a heat map over the anatomical MRI, and this is what we communicate to the surgeon. But rather than, so there we use the brain model as a template, but we can use it also as a in silico platform to run autonomous simulations. Here, just as an example, the blue dots represent the sources, the individual network nodes. And when there is lots of energy in terms of discharges in there, you see these red balls, these red spheres coming up, and SEG via the yellow spheres. So this is where you actually sample the activity, which I'm plotting down here for you. So as an in silico platform, we can now use it autonomously to generate seizures and to mimic interventions, perform surgery, to take different surgical approaches, Rather than performing a resection, we can perform a disconnection or interrogate different hypotheses for this patient. What we are doing, we are running a clinical trial right now. We are building patient cohorts, and the most advanced application is actually where we use the virtual brain as a template for fitting, because it mixes nicely these causal mechanisms and allows you actually to extract a informed statement about how likely it is to contribute to the epilepsy that we are seeing. You see an example for a successful fitting seizure-free patients. So overlaid, the black areas is where the resection has been performed, and in red is where we made our predictions coming from the virtual brain. Here you see one that was less successful. This is a non-seizure-free patient, and you have a deviation actually between the surgery that has been performed and actually the prediction that came from the virtual brain. So we are running this with about 400 patients. The clinical trial started in 2019. Involves 13 reference centers of epilepsy in France. The data are being centralized in a database, anonymized, and being sent to Marseille, where we actually virtualize the patient, perform the type of analysis that I described to you, and then we send a clinical report back to the various reference centers, where the system then, where this information is then being taken into consideration for the decision making. Half of the patients, of the 400 patients, receive this information, or the clinicians receive this information, the other half not, which will allow us to have these two arms, a VP positive, VP negative arm, to be able to generate statistical power. We need 400 patients. In December, we had 210 patients. Today, we have around 270. I don't have the graphics for that. So we are on track. By next year, 2023, I think we should be able to finish the clinical trial. The first results are promising. This has been done on retrospective patients. Here you have clinical hypothesis, and VEP, this is virtual epileptic patient coming, the prediction from the virtual brain model. Precision, but we have other statistical predictors also. This year's precision, and you see for 12 seizure-free patients, how the precision actually drops down as compared to the surgery that has actually been performed. And here you see some statistical quantifiers: precision, recall, APS, AUC, F05, so different F values. So the values are more or less promising. The VP is actually performant on a level that is comparable to the current state of the art in epilepsy. Just a quick use case for illustration. Here we have had a particular patient that was that went through clinical routine, and in 3T we did not see any lesion. It was a completely MRI negative patient, but you see actually when you go through high field MRI, there is actually a lesion that has been detected, a dysplasia in the left orbitofrontal cortex. In 70, it was visible. The patient was implanted, and seizure recordings were performed, in particular also some of them around the lesion area that has been seen. High resolution representations here in high resolution EEG, some model inversions, so source reconstructions were performed that did not correspond too well with the dysplasia in one. When you did this simultaneously with high resolution EEG and MEG, the fit was better to some degree. And actually with SEEG, there were different markers that were employed: spike rates, an epileptogenicity index, which is a ratio of high frequency discharges, mostly in the gamma band, to low frequency discharges, mostly in the alpha and beta band, is also used as a biomarker. And connectivity epileptogenicity index takes also the connectome into account. So here in these cases, the clinical conclusion was that two areas are implicated: left orbitofrontal cortex and left insular gyrate brevi were implicated. From the VP, we had in addition also the left inferior frontal sulcus that was indicated, but was not taken into consideration. Yeah? Surgery was performed, taking the two areas out, resected brain regions I just described, left insular gyrate, left orbitofrontal cortex in particular. So there was not a full overlap. And actually the patient, after 1 year follow-up, it was angled glass, so it was not seizure-free. Yeah. And so we went back into this one particular patient and focused on this one area that was actually identified by the virtual epileptic patient, and went into the MEG and zoomed into these areas in order to see if there is some activity. And there was actually some activity. Some interictal spikes were identified in these areas. Yeah? So that allowed us, after the fact, essentially to zoom in and identify the areas, and the patient now is being considered for further surgery for the left inferior frontal sulcus. So you can, once you have these personalized brain models, you can actually go in very much details and make converge and fuse the individual modalities. Yeah. So quick outlook of what is happening now. You can actually go to very high resolutions. The standard TVB today has about 200 areas, which gives you a coverage of about 10 to 20 square centimeters, which is huge. You want to go in more detail because just already the improvement from source to sensor mapping, where you can take the double momenta into account, will be a huge step forward. For this, we use actually within the Human Brain Project and eBrains, high resolution data that are coming from ex vivo brains. Here you see Roxana Coleman's, who's preparing the brain for imaging that allows us to go on a level of about 20 micrometers, in fact, approaching cellular resolution. Yeah, we need good projections and nonlinear spatial transformations, mapping the various data because they come from different sources into the same reference space. This is actually key to be the use of ex vivo data. Here you have the Chenonceau brain, where the brain that Roxana was operating on was scanned during 2 months with a resolution of 200 micrometers. And here from an example of polarized light imaging that allows us to go down even to 1 micrometer. So it does improve systematically the resolution, because now we can go to a 1 square millimeter resolution, as you see represented here, merging the data, improving the dipole orientation and the source to sensor mapping, because now we can take dipole orientation into account, and improve systematically our forward solution. It allows us then to run high dimensional seizures, not only in one single neural mass, but actually mimic the type of experimental data here from Kathy Chavon's lab, essentially having propagation also within the epileptogenic zone, and having a detailed spatio-temporal organization. So this is just an outlook to show you where we can go. Here you have the rhinal cortex identified, projecting into other areas. When we put an epileptogenic zone area there into the rhinal cortex, you see activity arising, propagating, not continuously propagating, but then recruiting other areas. This very classic wave propagation that you see in that area once it has been undergoing the bifurcation into the epileptic seizure. So we can also now continue from the cortical areas into the hippocampus. So take the different CA1, CA2, CA3 regions into account, that is going into the wrong direction, and extend that, allowing us to actually model also the wave propagation in the hippocampus in a much higher resolution than was possible before. So this is the state of the art. It's not being clinically used yet, but essentially this is where we want to carry it, because the idea is to have digital twin technology that allows us to take the real brain in a variety of complex scenarios to mimic it in vivo, using in vivo and ex vivo data, mimic it in silico, test interventions, and also expand it to other scenarios, taking different type of stimulation data and contexts and task contexts into account to feed it back to guide diagnosis and therapy. That is a vision that has been formulated here in zenodo.org. Please check it out. Yeah, just look up the Human Brain Project and on this particular platform. So I wanted to give you an overview, quick overview of what can be done. I hope it was useful. These are my colleagues and friends that have contributed to this. And these are the funding mechanisms that we use. Thank you very much and bye-bye.